Hello everyone. Today, I'm honored to walk you through a review of engineering bacteria for medicine, delivery, diagnosis, and therapy. Genetically engineered bacteria, GEB, are a new biotherapeutic platform with a modular design enabling high biocompatibility and programmability. They show promise in treating tumors, IBD, metabolic disorders, and complex infections. Key modules include sensing, payload release, and biocontainment, but challenges such as limited clinical strains, functional trade-offs, and biosafety hinder translation. Future progress will rely on precision gene editing, smart responsive systems, and multidisciplinary cooperation to move from preclinical to clinical use. 1. A unified modular framework, sensing processing actuation biological control, is proposed to systematically conceptualize genetically engineered bacteria, GEBs, across diverse disease contexts. 2. It reveals the shared engineering logic underlying diverse applications, enabling cross-disease comparison and a rational design of GEB systems. 3. Key advances in genetic circuits, secretion systems, and metabolic reprogramming are integrated to highlight the programmable and dynamic therapeutic capabilities. 4. A systematic analysis of the key barriers to clinical translation is presented, including manufacturing consistency, host variability, immune interactions, and biosafety concerns. Over the past 50 years, bacterial engineering has evolved from protein production to living therapeutics, marked by gene transfer, 1973, recombinant insulin, 1982, CRISPR, 2012, chassis optimization, 2016 to 2021, and clinical theranostics, 2022 to 2025. Future integration of synthetic circuits and AI will enable smart living drugs that combine delivery, diagnosis, and therapy. GEBs are genetically modified microorganisms that express specific functional proteins or execute programmed metabolism through the introduction of heterologous genetic elements. Their construction for medical applications follows a 6-step workflow: host strain selection, functional gene acquisition and codon optimization, gene deletion and genomic integration, synthetic circuit design and optimization, strain encapsulation and controlled cultivation, and multi-level in vitro and in vivo validation. Commonly used chassis include Escherichia coli, Lactobacillus, Salmonella, and Bifidobacterium. GEB-based drug delivery systems integrate synthetic biology and nanomedicine, offering significant advantages in treating tumors, metabolic disorders, and inflammatory diseases. The platform can be abstracted into 4 core modules: a microenvironment sensing unit, detecting pathological markers and enabling precise targeting, genetic logic control circuits, Boolean logic gates and genetic switches, spatiotemporal drug release mechanisms, condition-triggered precision delivery, and biocontainment strategies, auxotrophy and kill switches for safety. This modular framework reveals shared engineering principles across diseases, guiding the rational design and optimization of GEB platforms. GEB therapies span oncology, homing/lysis, immunogenicity limits, IBD, anti-inflammatory secretion, persistent safety balance, metabolic diseases, enzymatic correction, burden trade-off, infections, antimicrobial plus immunomodulation, multi-strain consortia, and diagnostics, biosensing/output. Despite shared modularity, each requires disease-tailored sensing, release, and safety strategies to balance efficacy and safety. This review outlines GEB evolution from biopharmaceutical hosts to living therapeutics, covering modular design and preclinical applications in tumors, IBD, metabolic disorders, and infections. Clinical translation is hampered by controllability, consistency, and safety issues, demanding a shift to level robustness. Future progress requires standardized chassis, reliable circuits, scalable manufacturing, and rigorous long-term safety/efficacy evaluation.